The central dogma illustrates the flow of genetic information in cells, the DNA replication, and coding for the RNA through the transcription process, and further RNA codes for the proteins by translation. In this video, we will guide you through the step-by-step process of the central dogma of biology, including initiation, elongation, and termination, for each step. Let's get started. DNA replication creates an exact copy of DNA. It occurs in the nucleus before a cell divides. The initiation point where the splitting starts is called the origin of replication. The helicase enzyme separates the strands by breaking hydrogen bonds. The replication fork forms the site where the new DNA strands will be synthesized, with the leading strand oriented 3' to 5' and the lagging strand oriented 5' to 3'. Topoisomerase relieves strain ahead of the fork. Single-strand binding proteins prevent DNA rewinding. An enzyme called primase creates a short sequence of RNA called a primer that provides a starting point for elongation. Next comes elongation. DNA polymerase III starts to synthesize new strands. The leading strand elongates continuously towards the fork. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as Okazaki fragments. When DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. The primers are removed and the gaps are filled with DNA polymerase I. Then, an enzyme called DNA ligase seals up the sequence of DNA into two continuous double strands. Finally, enzymes called nucleases proofread the new double helix structures and remove mispaired bases. DNA replication results in two DNA molecules, each with one old and one new chain. It's semi-conservative, retaining half of the original DNA. Next is transcription. It is the process by which DNA gets converted into mRNA, also known as messenger RNA, which carries genetic information from the nucleus to the cytoplasm. During initiation, a section of DNA called the promoter signals where and when transcription takes place. The protein RNA polymerase finds this promoter and separates the double-stranded DNA. One of the strands will serve as the template strand or antisense strand, meaning it will be used to generate the mRNA. And the other is the non-template strand or the sense strand. Elongation occurs as the RNA polymerase reads the template strand and builds a complementary RNA strand by bringing the appropriate nucleotides together. This RNA strand is almost identical to the non-template DNA strand, except that the thymine in DNA are replaced with uracil in the RNA strand. This continues until the entire gene is transcribed, resulting in the formation of messenger RNA. Termination happens once the RNA polymerase reaches the terminator sequence, breaking the hydrogen bonds between DNA and mRNA. Then, the RNA strand separates from the template DNA. The DNA returns to its double helix formation, and a newly formed mRNA strand is ready for synthesizing a new protein. The edited mRNA exits the nucleus through nuclear pores, enters the cytoplasm, and proceeds to a ribosome for the next stage, which is translation. Translation occurs as a complex is formed that is comprised of the large and small subunits of a ribosome, the mRNA and tRNA. The mRNA is read 3 bases at a time. These 3 base sections are called codons, and they determine the amino acid required. tRNA has a binding site, referred to as the anticodon, that contains the 3 bases that pair with the bases in the mRNA codon. During initiation, the ribosome recognizes and binds to the start codon AUG, which is usually the first codon in an mRNA molecule. A tRNA picks up the required amino acid from a pool of free-floating amino acids in the cytoplasm, and brings it to the ribosome. Here the anticodon and codon attach, putting the amino acid into place. During elongation, the ribosome again moves down the mRNA strand, a tRNA brings in the required amino acid, and the two amino acids form a peptide bond. This process keeps happening, forming a long chain of amino acids. At the termination phase, translation continues until a stop codon is reached. The polypeptide chain is then released from the ribosome, most likely entering one of the cell organelles for folding and further modification. And there you have it. From DNA replication to RNA transcription to protein synthesis, this process is essential for all living things. We hope this video has helped you better understand the central dogma of biology. Thanks for watching.